Thanks a lot for the kind introduction and for including me into this fantastic group of talented scientists here in the first place, and for making a tremendous amount of effort to get us here regardless of all kinds of lockdowns and such. In my graduate research in Daniel Mucida's lab, I studied the crosstalk between gut microbes, nervous and immune compartments in the gastrointestinal tract. So our body is constantly confronted with a large variety of foreign environmental stimuli, and while our immune system is in place to protect against dangerous threats, we also need to avoid excessive inflammation that can lead to tissue damage and remain tolerant to harmless or beneficial dietary and microbial antigen. Now, in our lab, we study these dynamics in a setting of the murine gastrointestinal tract, which, as mentioned, is one of the largest environmental interfaces of the body, and it contains both the body's largest immune compartment as well as a vast neuronal network, the enteric nervous system. And together, these two regulate homeostatic tissue functions, such as digestion, nutrient absorption, and secretion, while at the same time defending against potentially dangerous threats. Now, when we take a cross-section of the intestine and then zoom into just a small little insert here, we can see that it consists of multiple layers with just a single cell layer of epithelial cells that separates the body from the environment, including a community of trillions of viruses, protozoa, bacteria, and fungi that are collectively termed the gut resident microbiota. And then underneath, in a so-called lamina propria layer, we have the body's largest fraction of immune cells that's strategically placed to keep out pathogens. And then further down, embedded between muscle and stromal layers, we have the two plexuses of the enteric nervous system, the submucosal one and then the larger myenteric plexus. And for the purposes of today's talk, I'll be mostly focused on this myenteric region down here. Now, the intestine is a highly innervated organ, and this innervation consists of this intrinsic innervation and then in addition an extrinsic one. Now, the intrinsic innervation are the two plexuses of the enteric nervous system, and these are located within the intestinal wall, and they also have a unique feature, and that's their ability to operate completely autonomously from the central nervous system. And they're tasked with modulating intestinal motility, secretory and absorptive function, and then get additional aid coming from extrinsic innervation. Now, these are sensory afferents that transmit information from the intestine to the central nervous system, and then autonomous sympathetic and parasympathetic efferents that transmit top-down information from the central nervous system to the intestine to aid in fine-tuning intestinal function. Now, using a special technique that was actually developed at Rockefeller, where you can make a piece of intestine completely transparent and then image its components in a three-dimensional space. Now, here we are looking at a piece of distal small intestine, and we have neurons labeled in white. And I think you can really appreciate this very dense innervation of the intestine, what we find. And we have this web of neurons which lines the intestinal tube, and these are intrinsic intestinal neurons, but we can also see fibers reaching the intestinal wall, and these are extrinsic gut innervating neurons. Now, all these components, the microbiota, nervous and immune systems, communicate with one another, and this crosstalk was the major focus of my work in Daniel's lab. And specifically, we wanted to elucidate the influence of gut-resident microbes on the enteric nervous system and tissue physiology. And what we found was that there's a microbiota-modulated enteric neuron subset that autonomously regulates glucose metabolism. And in the second part, we wanted to determine the impact of intestinal infections on the enteric nervous system and gastrointestinal function. And here what we found and mostly focused on is that the crosstalk between intestinal immune cells and gut-associated neurons provides tissue protection during intestinal infections. To just give you a brief overview of this first part, we found that using translating ribosomal affinity purification to isolate intestinal neurons, we found that intrinsic intestinal neurons are a transcriptionally unique population of peripheral neurons. And we found that the gut microbiota has a strong impact on intestinal neurons, and that's specifically true for distal intestine neurons where there is a stronger microbial community. We found that specific intestinal neuron subsets are dependent on the microbiota for their survival, and then we defined a specific population of these neurons that regulate glucose metabolism via a circuit that's independent of the brain and connects the intestine, pancreas, and liver. We found that a healthy microbiota is necessary for the survival of enteric neurons, and that microbial depletion leads to the loss thereof. And so we then wanted to determine the impact that intestinal infections have on the enteric nervous system and gastrointestinal function. Now, enteric infection often results in gastrointestinal disorders after pathogen clearance, and one such example is post-infectious irritable bowel syndrome. And now here, primarily bacterial infections such as Salmonella and Campylobacter can trigger acute inflammation, which then persists long-term after pathogen clearance. And these symptoms include pain, low-grade inflammation, and nerve damage, and are found in a subset of patients with intestinal infections. So we then developed an infection model that recapitulates some of the hallmarks of post-infectious IBS, where we infect mice with an attenuated strain of Salmonella called SPI-B, to then explore gastrointestinal associated neuronal, immune, and functional changes triggered upon infection. SPI-B is cleared about 1 week post-infection, but it then leads to a persistent low-grade increase in fecal lipocalin-2, which is a marker of intestinal inflammation. And this peaks at around day 6 post-infection, so before pathogen clearance, but it then persists at later time points. And in addition, we also find an increase in a so-called total intestinal transit time, which is the time that it takes for a test meal to travel through the entire GI tract, and is a measure for gastrointestinal function, where an increase in this time can be used as a proxy for constipation. And then importantly, we also found damage to the enteric nervous system, as we found a loss of about 30% of the neurons in the myenteric plexus region of the distal intestine. And this was not only true for Salmonella, but we could recapitulate these findings in other both bacterial as well as protozoan pathogen models. And these changes also persisted long-term after infection, which was reflected both in a persistent increase in intestinal transit time and a persistent loss of intestinal neurons even months after infection. So we see that in the absence of further perturbation, there seems to be little to no tissue recovery. However, we also noted a shift in the microbial composition after SPI-B infection by 16S bacterial sequencing. And we then wanted to see whether, if we treated mice with antibiotics to clear out their intestine and then perform fecal transfer, giving them back a pre-infection or a healthy microbiota, whether that could induce neuronal recovery. And indeed, as compared to mice that only received Salmonella infection, here the gray line just indicates normal neuron numbers. So we see neuronal loss upon infection, but when we treated mice with fecal transfer and normalization of their microbiota, we found complete neuronal recovery, indicating that dysbiosis hinders and the healthy microbiota enhances neuronal recovery. But now, what is the mechanism behind this neuronal loss? In other words, how do these 30% of neurons die? We used RNA sequencing of neurons that are located either inside the intestine or gut innervating extraintestinal neurons. And what we found was that as compared to extrinsic neurons, intestinal ones had highly and selectively expressed NLRP6, which is a key member of the inflammasome pathway. And here activation of this pathway leads to a cell death mechanism via pyroptosis, so an inflammatory form of cell death, via the caspases 1 and 11. And importantly, these intestinal neurons also express the downstream machinery of this pathway, so caspases 1 and 11 and the inflammasome adapter protein piCARD, showing that these intestinal neurons possess the machinery to die in an inflammatory manner. To then see whether they actually do that, we used an intersectional genetics approach where we specifically deleted NLRP6 from neurons. When we then subjected mice to Salmonella infection, we found that as compared to mice that had functional NLRP6 and showed normal neuronal loss, mice that lacked NLRP6 specifically in neurons showed complete protection from neuronal cell death, indicating that this infection-induced neuronal death is indeed mediated by neuronal NLRP6. Now, what prevents further neuronal death post-infection? We know from previous work in our lab that there's 2 intestinal macrophage subsets that reside either up here in the lamina propria layer or down here in the muscularis layer, and these have different gene signatures and functions in protective immunity. And specifically, this work showed that these lamina propria macrophages are more pro-inflammatory, whereas muscularis macrophages are more anti-inflammatory and tissue protective in nature. And these muscularis macrophages are also in close contact to and continuous communication with the neurons of the enteric nervous system. Now, given that they're the only immune cell type that's present in abundance in this neuronal layer, we wanted to determine the role of these macrophages during intestinal infections. So here on the right, we can just see that macrophages labeled in green and neurons in red, and we can see this very close association of these 2 cell types. And we now wanted to see what happened when we disrupt these interactions, and for that we used an antibody to selectively deplete muscularis macrophages that did not impact lamina propria ones, and to then see what happens when we disrupt these interactions in the context of an intestinal infection. We first looked at steady state where depletion of macrophages did not impact neuronal numbers, but when we did the same over the course of an infection, we found further enhanced neuronal damage. indicating that these nerve-associated muscularis macrophages play a role in keeping neuronal damage at bay. So now to summarize this second part, we found that different intestinal infections, but foremost salmonella, leads to an impressive loss of intestinal neurons. And we find that intestinal muscularis macrophages rapidly respond to these enteric pathogens to limit neuronal damage. And I didn't have time to show you the specifics here, but we see that this protection involves adrenergic signaling in arginase 1 and polyamine axis, where the secretion of these polyamines from macrophages leads to a blocking of NLRP6 in neurons. And finally, we found that post-infectious neuronal cell death is mediated by a novel mechanism that involves the NLRP6 inflammasome and caspase-11. We found a strong influence of the gut microbiota on the enteric nervous system, and we found that microbial disruptions lead to an inflammatory form of neuronal cell death. And then we had 2 examples of what happens when we disrupt the interactions between gut microbes and neurons. At the steady state, we see that the loss of a microbiota-dependent neuronal population that feeds into a central nervous system independent neuronal circuits leads to disruptions in glucose homeostasis. And we see that the loss of an intestinal neuron population during intestinal infections leads to gastrointestinal dysfunctions. And finally, we see that an intestinal macrophage population is critically involved in preventing excessive neuronal damage. And with that, I would like to thank again the organizers for inviting me to present this work. And I would like to thank all of the members of the Mucida lab, especially Daniel and my collaborator, Paul, who was a co-first author on both of the papers. And yeah, thanks for your attention.